There are 3 levels at which we can measure biodiversity: habitat biodiversity, species biodiversity, and genetic biodiversity. Now, habitat biodiversity is the range of different types of habitats in an area. And now the higher the habitat biodiversity, the higher the species biodiversity. For species biodiversity, we can consider both the richness and the evenness. So we have species richness and species evenness. Now, whilst richness is simply the number of different species, the evenness refers to the relative proportion of individuals of each species in a habitat. Now, Simpson's index takes both of these into account, and this is the formula. Now, the result of the formula will always give you an answer between 0 and 1, with 1 being a very high species biodiversity and 0 being the lowest. So essentially, for any given ecosystem, we can do this calculation and your answer must always fall in between 0 and 1. If you're ending up with negative numbers or numbers greater than 1, then you've done something wrong. So what I'm going to do is I'm going to show you an example. There we go. So consider 2 fields in which both of them have clover, dandelion, and daisy. They both have the same species richness because both fields have only 3 species of plants. However, they have different evenness. Now, as you can see for field B, the numbers are more even. So if we were to draw these on a bar chart, the heights for this field would be a lot closer together. So this means that field B has a greater species evenness. Now, as the formula takes both richness and evenness into account, the field with the highest richness and evenness would have a greater Simpson's index of diversity. So since they both have the same richness, this one has a greater evenness, B would have a greater Simpson's index, for example. Now, what we can do is we can actually work this out just to prove that this is correct. Now, one thing we can do to help us work out the Simpson's index of diversity for field A is we can draw a table just based on the data for this field. So what we've done is n being the individual numbers and n over big N, this big N being the total. And what we have to do is we have to square everything in this column. Subtract this from 1. Which gets you to an answer of there we go, which is actually a very low value. It's very close to 0, indicating that this field has a very low Simpson's index of biodiversity. This field has a very low species biodiversity. It's very close to 0. Now, if we were to repeat this calculation for field B, which you can try, I'll put the answer in the comments, species biodiversity is going to be a lot higher, as we have predicted just by looking at the relative proportions. You can see that this is a lot more even. The genetic biodiversity refers to variation between individuals of the same species. Now, you can measure this using the percentage of polymorphic gene loci, and the higher the percentage, the population can be considered to be less susceptible to disease. For example, as within this population, the advantageous allele is likely to be present, as there are a large number of polymorphisms present. You can work this out by using the number of polymorphic divided by the total number of loci times 100 as it's a percentage, and this will give you the percentage of polymorphic gene loci, and the higher the better, essentially. The higher the genetic biodiversity, that's basically better. When studying ecosystems, we have to do a lot of sampling. Now, sampling can be split into both random and non-random. Now, random is exactly as expected. Every member of the population has an equal chance of being selected. Now, non-random is exactly the opposite. And non-random sampling can be split further into 3 different types. So we can have opportunistic, which is probably like the most easy kind of sampling. You just do it based on convenience, whichever is easiest to do, whichever organism is easiest to be sampled, for example. Stratified, which involves splitting the population into strata and then drawing samples from the subgroups. Now this is good because it makes sure each subgroup is proportionally represented in the overall sample. And the last one is systematic, is where we take data at regular intervals, for example, every 10 meters. Now a quadrat is a frame of known area, usually 0.25 meters, so this can vary, and it's placed randomly within a habitat to estimate a bunch of things, including abundance, distribution, and species richness. Now normally, random sampling is used because this avoids bias. What we can do is we can split the field using a coordinate grid, and then we can use a random number generator to decide where to place the quadrat. Now this is used to sample plants and slow-moving organisms. We can also use transects along with systematic sampling to investigate how biodiversity changes with distance. In this case, as we are investigating how biodiversity will change, systematic sampling has to be used. For example, if we were to measure the distribution of a certain kind of plant, along like a coastline, for example, we take readings every 10 meters or so, for example. Next up is sampling animals. So now we're going to talk about techniques used to count the number of animals, and this will depend heavily on the type of organism. For example, we can use something known as a pitfall trap. And this is for crawling ground insects. We can use a pooter, which is where you basically suck insects. Also for crawling ground insects. A Tallgren funnel. Which is for small organisms which live in soil or leaf litter. So basically the process for this is leaf litter is put on a mesh filter at the top of a funnel, and then light is shone down on it, and organisms will basically move away from the heat that's created by this light source, and they will fall out of the funnel and into a beaker. As you can see, this will only work for small organisms. Next one is kick sampling. And this is for aquatic organisms. What you do is you disturb the bottom of a stream and then use a net to collect organisms, and lastly a sweep net, there's no space to write this, which is for organisms in the long grass. Basically just a net. Now, one thing they like to ask is why maintain biodiversity? Well, there's a number of reasons, but they can be split into 3 categories. So there's ecological reasons. Okay, now the next one is economical or economic. Now, a lot of medicines and pharmaceutical products are derived from things found naturally in the wild. And lastly, aesthetic, as simple things like walking in nature are known to be very good for your well-being. Okay. One thing I need to mention here are these acronyms, which are basically treaties or schemes that have been put in place to increase biodiversity. So CITES stands for the Convention on the International Trade in Endangered Species, which was basically put forward in 1973 and was signed by 145 countries, which is a treaty that prevents the trade of endangered species, basically. And this one is the Rio Convention. but first I'm going to explain what CBD is. It began in 1992, and countries had to come up with strategies for sustainable development and to stabilize greenhouse gases, for example. And basically, this allows the sharing of access to scientific knowledge and technology. Now, the CSS, or the Countryside Stewardship Scheme, is UK specific, and basically, landowners were paid to set up things that would protect and enhance the natural environment.